In this session, we will look at how to fine-tune a protein large language model. To find the relevant tutorial, first we go to the training webpage of Galaxy, which is training.galaxyproject.org. And then we find the right category in which the tutorial is located. We go to statistics and machine learning. Then we click it, and then inside this category, we will go a bit down and then find fine-tune large protein model ProtTrans using Hugging Face. This is the tutorial that we will be looking at to do hands-on, in which we will fine-tune a protein language model using dephosphorylation datasets. Let's open this tutorial. The learning objectives of this tutorial is to learn how to load these large language models from Hugging Face, and then how to fine-tune them on specific data sets. In this hands-on, we will use dephosphorylation site prediction task and the protein sequences that are known to be dephosphorylated and not dephosphorylated. So there are two classes to the protein sequences. But the tool that we will be discussing in this tutorial, using this tool, multiple downstream tasks can be performed using large language models. This is just an example. Let's look at this tutorial in detail. We discussed in the previous session that researchers have created a biological version of large language models by training them on biological data sets such as DNA and proteins to create models such as ProtBERT, DNABERT, ProtAlBERT, and so on. These pre-trained models trained on protein sequences and DNABERT, they contain the knowledge about the context of the protein sequences and DNA sequences. And using these pre-trained models, we can either extract the embeddings for our sequences, and using these embeddings, we can train a different machine learning model to find classification accuracy. Additionally, these pre-trained models can also be fine-tuned on the new downstream dataset. It is expected to give better accuracy because the pre-trained model contains a lot of knowledge about protein and DNA sequences. And the representation that is learned by these pre-trained models is better compared to the ones learned by any machine learning model which starts with a random weights. For this examples, we will use ProtT5 model trained on UniRay 50 dataset to fine-tune on dephosphorylation dataset. It will be a binary classification task. First of all, let's briefly look at what are dephosphorylation sites. Dephosphorylation, as mentioned in the previous session, is a biochemical process which is commonly known as post-translational modification of a protein after it is synthesized. In this process, a particular compound gets attached or detached from a protein molecule, and because of this event, it regulates the protein sequences in a different way that could lead to diseases if it is not normal. For example, if we remove phosphate groups from proteins, then it can interfere with with the enzymes, and it can either activate it or deactivate it, and that could lead to different problems in cell growth and so on. And they are known to be associated with diseases such as cancer, diabetes, and different neurodegenerative diseases. To predict whether a protein sequence contains a dephosphorylation site or not, first we will download the pre-trained model from Hugging Face. Then using low-ranking adaptations, we will extract a low-ranking model of the original pre-trained model containing very few number of parameters. So we will go down from 1.2 billion parameters in the original pre-trained model to 3.5 million trainable parameters in a low rank adaptation of the original model. And we use this low ranking model to fine tune it, and then we will predict it on the test data set. First of all, we will use a general purpose tool in Galaxy, which is a GPU enabled. This is an interactive development environment, popularly known as Jupyter Lab. And this is hosted inside Galaxy Europe. And users can use this notebook to write programs. And the programs written in, for example, PyTorch or TensorFlow can run on GPUs, and this ensures faster training. First of all, we will start a Jupyter notebook. To start a Jupyter notebook, users need to go through an access mechanism. Here at the bottom, we specify a Google form through which users can get access to use this tool. This is a very simple form and that can be filled, and it takes a few days to get it approved by us, and then users can use this tool whenever they want. So it asks for an official email ID and the institutions, and you can provide a short description of your use case, and then few other details, and then you can submit. We will get the request, and in a few days you will get this access. We have implemented this access because Jupyter Notebook accesses GPUs, and we want to be as fair as possible for all users and make it equally available for everyone. So, and to prevent abuse of this resource, so we have put as an access mechanism for this resource. Therefore, because this resource is not directly publicly accessible, this tutorial and hands-on session will be like a guided example or guided hands-on, where I will show you how you can use this tool to fine-tune your favorite protein language model using your favorite downstream task. When you have access to this tool, then we go to the Galaxy and copy the name of the tool. Let's find this tool in Galaxy Europe. First, we copy the name of the tool, and then let's find this tool in Galaxy Europe. Then we found it as the first tool here. And then we open it. Then here we can see that our GPU-enabled Jupyter Lab notebook tool for machine learning is open. And then we are currently using version 0.4. Then it asks for certain inputs from our side. In this dropdown, we can see there are three options. Start with default notebooks, then the container in the backend, which opens this notebook, has certain notebooks available that shows the features of this notebook. Then these notebooks become available. We can also start this notebook with a code repository from Git. For this, when we choose, then we have to provide a URL to the code repository. For example, one example is given here, that we put it here, and when the notebook environment opens, then it will automatically be created and downloaded inside. Then we can also run it, a notebook that is supplied as an input. Then this tool works as a regular tool in Galaxy that executes our notebook and provides the output. To do that, we have to supply the notebook to be executed, and then we have to select this flag here, execute notebook, and return a new one. Then it will not open a Jupyter Lab environment. It will just execute the notebook and provide the outputs in the current history. We can also insert user inputs. For example, if we check here, then we have to provide a name for the folder which will be created inside the notebook. And then we can include different types of data set, which can be controlled by here. For example, if you want to include a text data set or some other text data set, a Boolean data set, and so on. So we can just choose it from here and then just run this tool. I have actually executed this tool, which is running. When your access request is processed, then you will be able to run this tool also on your own. When the tool is running, it shows something like this. It has a URL. This URL, you can send it to your collaborator, which also sees the same thing as you are seeing, just like a Google Doc. If you remember, we chose the option of cloning a repository at the start. We did it, and then our clone repository is present here in this folder. We go to this folder, and then we find here one notebook and two FASTA datasets. These FASTA datasets are training data, training protein sequences along with the labels, and the test protein sequences along with the labels. We can maybe have a look here. The FASTA looks like this. So here is a protein sequence, and the middle nucleotide in this sequence is the dephosphorylation site. And this is a positive site that is determined by label 1. Then we can also find like some sites which are also negative here with label 0. And test sequences also look the same. They have label 1 and label 0, but the number of test sequences is only 100. It's actually not 100, but is actually 50, because each FASTA sequence has 2 lines here, one for the special information and one is the sequence. So there are 100 lines. You have 50 sequences with labels. We go to this notebook and see what are the contents inside this notebook are. We a little bit go to the left here. We can see that we need to install some packages. The Docker container that is running behind this Jupyter Lab notebook contains a lot many packages for machine learning and data science projects, but it doesn't contain all of them. It by default it contains TensorFlow, scikit-learn, scikit-image, OpenCV, and so on. And then the CUDA packages are there to interact with GPUs, but it doesn't contain PyTorch related packages. Since the pre-trained model has been created using PyTorch, and it's available from Hugging Face using Transformers package, we need to install PyTorch and all relevant packages. So in one uh, install statement here, it installs all the necessary packages. So we are just going to run this cell. We started running it and we see the packages are started getting downloaded, and it takes like a minute or so to download all the packages. Then we see the Torch package is getting downloaded. When the download is complete, then we can actually import all these packages. So similarly, when you are working with your favorite protein language models, then you can also download the packages that are relevant for your project. Or you can follow the same tutorial and see if these set of packages also work for your downstream tasks. So it still needs to download a few more packages. So in the meantime, we can see some of the features of this notebook. One such feature is Git. So you can download any repository from Git and make some changes in the notebook, and then also you can push your changes to remote repository using your API key or GitHub password. This feature is integrated in the notebook itself. Then we have dashboards to look at the GPU utilization. So we have certain dashboards, some charts to see CPU and GPU utilization, which is handy in some cases. So when we start utilizing the GPU, when we are training the model, then we see the utilization going up. If you like to learn different features of this notebook environment, then you can go to the notebooks folder, and that opens when you run this tool. And then you can find different notebooks written where you can explore different packages. For example, you can create your models to Open Neural Network Exchange format using model sharing. Then you have different visualization notebooks, how you can visualize your data using different techniques. Then you can also connect to your Galaxy data sets and workflows and tools using BioBlend. It's connected. Yeah, we just see whether the installation has finished or not. So it looks like the installation has finished, and I think it's not finished yet. I think it needs some more time. So in the meantime, we start running the next cell, and once it's finished, maybe the next cell will start running. Okay, so this has finished, so we can run the next cell. So you see different packages will be imported and all the versions would be listed here. This notebook contains several sections. First is actually installing all the packages. Then we have to fetch the data from FASTA files and do a little bit of pre-processing. So now you see all the packages have been installed, and you can see the versions. Torch version is 2.1.1, which is also a CUDA version. It will automatically recognize GPUs and start using it. And we also see that the transformer package version is 4.35, which will be useful for downloading large language, pre-trained large language model from Hugging Face. Let's try to access the data set here. So the data set looks like this. We have a sequence identifier here, and then we have a sequence, and then the labels for the sequence. This shows the top 5 values from the data set, from the training data set. Then here we split the data into train, test, and validation set. So the training is only 160, and the validation set is 40. In the previous session, we discussed about low ranking adaptations of a model, using which we can divide the weight matrices into smaller matrices. And these smaller matrices contain all the knowledge of the downstream data set when the training is finished. And this low ranking representation, so the matrices are added back to the original frozen matrix of the pre-trained model. And then we have only a fewer number of parameters in the training model, and which makes it faster. So all this script is written. You can see that the rank of the new small matrix is only 4, and we are actually applying the lower rank only on linear data sets, linear layers in the model, which is specified in the script here. Then we have to modify the original model with LoRa and apply the LoRa matrices everywhere, which is happening here. Here, this class defines the T5 architecture of the encoder. Since we are using only the encoder and not the decoder part, because we just want to learn the representations of the input sequence and then map it to the class labels. Then we go down, and then here now we will download our protein large language model from Hugging Face. So we saw that this is the transformer package, and this model will be downloaded from Hugging Face using from pre-trained method. Then we have to provide the name of the model that we are downloading. So this is a key to the model, which is the unique ID of the model. And then we provide it for the model and its tokenizer. And then the entire model gets downloaded. It needs some time. So I think we forgot to run these cells. Let's run them also. Right. They will be utilized a bit later. So we have model here. Here we set some functions that creates PyTorch data sets using our label data, the training sequences and the label data. Here the tokenizer, you see the maximum length is 1024. Then we specify we are training per protein, like per protein, it's learning the representation for each protein. Then we provide training and validation data. Then we have only 2 labels, 0 and 1. then this is the batch size of 4. So every time it takes only 4 sequences and then makes classification, accumulates the error, propagates it back to the model, improve the weights, and then takes next batch of 4 sequences. For validation, it uses a batch of 16. And then we go on. Here we load the model. We have our LoRa models, and here we specify the training arguments. What are the training arguments? What should be the batch size? What should be the validation batch size? And what are the number of epochs? And so on. Then we specify the metrics. What are the different metrics that are needed to evaluate the accuracy of training and validation? Then there is a trainer method. Using this trainer method, we can train the model, or it will be actually fine-tuning the model. Then we just run this block. And here in this cell, we are actually training the model. We see that our batch size is actually 1. We are training per batch 1. And then the epochs is 20. Then we just run this. Then first of all, it will download the model. We see that the model is 11.3 gigabytes. It takes maybe 1 or 2 minutes to download this model. And once this model is completed, the download is complete, then the training will start. Then beyond the notebook, so we have different scripts written for analyzing the results. And we analyze it by looking at the training and validation loss values and also the confusion matrix to find if the model is performing well on training and validation both. So here, this was generated previously. So when we run it for 20 epochs, so we see that the training and validation loss go side by side. They are not too much different, which says that the model training is stable. This script provides a way to save the model locally, right? And then using the load model function, it can reload the model, and then it can be used for different purposes. We also look that the original model, the PROT-T5 classifier, is 1.2 billion parameters in size, but the same model with a low ranking adaptation, it has model parameters around 3.5 million. Let's see if the model download is complete. I think the model download is complete. The training might start soon. In the meantime, we just run all these cells. Let's look at the confusion matrix that was plotted before. So here we see that maybe I will zoom out a little bit to show this confusion matrix in full. Here we see that on y-axis is the actual labels, on x-axis is the predicted label. And we see that the actual model and the 20 here, which are from the 0 and also predicted as 0. And there are 17 examples which are 1 and also predicted as 1. So for a good classification performance, the values in this cell and this cell should be higher compared to the off-diagonal elements. It says that out of 50 test examples, 37 has been predicted correctly, and 13 has been predicted in a wrong way. Let's go back to the tutorial and see. We can see that the training has started. Here we already see epoch 1 is completed, and the training loss is 0.72, validation loss 0.68, and accuracy is 0.56. And for 20 epochs, it has 400 training iterations. And then we see that the accuracy increased in the second epoch and so on. It takes about 20 minutes to finish 20 epochs. I was talking about GPU utilization. So when we go to the dashboard of GPU utilization, we can see that GPU utilization is around 100% here. We can also look at different dashboards, how much memory is being used. You can see that it's actually fitting, the entire training is fitting on 7.4 gigabytes. It's taking only 7.4 gigabytes, which is like 50% of the entire GPU. You can also see the GPU utilization here as well. It's close to 100%. The training is happening. So using these dashboards, you can explore the utilization of your GPUs. You can see the training is progressing here. Once the training is finished, then we can see the plots like shown in the tutorial here, and also showed you these plots, which is saved in the notebook. When you go to the repository here that we cloned when we started the notebook, you can find all the saved results in this repository as well. Not waiting for the training to finish, so we can conclude the tutorial. So here in this tutorial, we saw a guided hands-on where we can use a GPU-enabled Jupyter Lab environment tool in Galaxy Europe to fine-tune a protein large language model, and to show that we can use a pre-trained large language model on protein sequences. We can actually do classification on a downstream data sets. This shows an example that is possible in Galaxy, and you can bring your own data set, not necessarily dephosphorylation, but for example, if you want to predict some protein classes, so you can bring that data set and you can follow the strategies of downloading the pre-trained model from Hugging Face using the low ranking adaptations. Sometimes you may need to do quantizations of the model to fit it into your GPU, and then you can perform downstream tasks. And then you will have some accuracy measured while fine-tuning large language model, and then you can compare it with other standard machine learning methods, whether it improves the accuracy or not. With this, I would like to conclude the tutorial. I hope there is some takeaway for you, and you can bring your own data sets and fine-tune a large language model which is suitable for you. We also ask you to provide your feedback. So we have a feedback section here at the end of the tutorial. So your feedback would be very useful to improve the content. So you can write Whether you like the tutorial or you did not like the tutorial, you can suggest us what we can add to the tutorial or what we can remove in the tutorial so that the overall learning experience becomes better. Thanks a lot.